Okay, thank you, Alex, for that introduction. So I'm actually going to talk about kind of one area of work in our lab, which is kind of a project that we've been working on for about 3 or 4 years, which has to do, it's an area called fine mapping, causal variance, and allelic heterogeneity. So here's actually our lab has actually pretty broad interests, but I think this work is something we've been interested in a while, but also gives a flavor of the kinds of things that we're interested in. So here's a summary of GWAS. So genome-wide association studies is a technique where genetic information was collected from case and control for individuals that have disease, individuals that don't have disease, and we compare all the frequencies of variance between those two groups. And if we see a difference in the frequency in let's say if a variant is more common in individuals that have disease than individuals that don't have a disease, that variant is associated, and that gene that variant is in may be a causal gene in that disease. And so this technique was applied very successfully over the last 10 years, and literally thousands of variants have been implicated in dozens and dozens of common human diseases. So this is very successful. And the results of a genome-wide association study is typically visualized by that plot over there, which is called a Manhattan plot, that on the x-axis has the coordinates on the genome, and the y-axis is some kind of a measure of how significant that association is. So the peaks there, they're labeled by the genes that they're in. It represents a finding that that gene might be involved in disease. So what we're interested in is a very straightforward problem, which is to say, okay, so if we know that there's an association peak at a gene, what is the actual variant that's driving that peak? That's the problem that we're interested in, and that's what's called fine mapping. So going from an associated region or associated, if you see that there's many oops, that was not good. So there's many variants that are kind of all along this peak. So the question is, which one of them is the one that's actually driving? So fine mapping has generated a lot of interest. There's been many papers on it, both application of fine mapping to understand, to GWAS peaks as well as methodology. In our view, we see fine mapping is actually really, we are interested in a set of related questions. So we're interested in kind of the core question, which is, if we have a association peak, what is the variance that actually causal? So what are the variant that has a biological effect and affects the phenotype? That we call fine mapping at the variant level. One level up is which genes are causal. So the variants, they affect which genes, and how does that affect downstream phenotypes. Then other related questions, if we know that are natural, if we know what the variants are, is how many causal variants are there in a locus? Is the signal that we're observing, is it just one variant or is it more than one variant? Fundamentally, if we're in the population, if there's a gene that's affecting a disease, is there only one variant that's causing that, or are there more than one variant that are independently causing that or interacting? That problem is called, or that phenomenon when there's more than one variant, is called allelic heterogeneity. And then finally, another related question is, if you have two studies, is it the same actual variant that's causing basically has the causal variant in both studies? So that typically this is applied if you have a genome-wide association study and also an expression, like a study of understanding which variants affect expression, and you want to know, is the same variant that's affecting expression the one that's affecting the disease? And that problem is called co-localization. So all these problems we see as closely related. So, okay, so why is fine mapping hard? Okay, so here we have the result on the top. We have the results of a GWAS study, and so there's a really nice peak that shows, that is clearly significant. However, when we zoom in, what we see is we see something like this. This is kind of zoom in to this point. We see that all of these variants are significant. And so it's very hard to tell which variant is the actual causal one. It's probably not all of them because there's a correlation structure in the genome due to the fact that we came from a small population in Africa a long time ago. And so there's, there's some correlation between who carries these variants that causes there to be many signals. So the question is, it's probably one of these or maybe a few of these. Clearly, it's not all of these variants are causing disease. So that's the difficulty. And what people currently do, the most widely used approach for fine mapping is they look at this plot, they look at it, and they kind of figure it out by hand. Okay, this is the standard, most widely used. Okay, I'll show you an example. Here's actually an example, right? Here, what they're also looking at is another thing. This down here shows the correlation structure. Correlation structure is often called linkage disequilibrium just for historical reasons. But what is this? This is actually kind of a correlation matrix that's on its side. And every point in this matrix tells you the correlation between this SNP and that SNP. And the color is the correlation. So for example, these dark colors say that these two SNPs highly correlated. This is all really light. There's almost no correlation that's longer, right? So if we see, and then here, this again is the height here is the strength of the signal. So if I showed you this example, and you were just using the standard approach of looking at the data, which SNP would you say is the causal variant? 33, very good. Yeah, very good. So that's the actual causal variant, very good. The way we know that is because it's from simulations. So we actually implanted it there, right? So that works there. But more often, the situation looks like this, okay? There's a lot of correlation, and there's a lot of significant associations, and it's very hard to see what's going on, okay? And there's really, so the problem we can think of is given the marginal statistics and given the results of the association study, as well as the correlation structure, how do we predict what the actual causal SNPs are? And again, the challenge is because there's correlation, and also there could be more than one variant in the locus. We'll talk about that in a second. And so the existing methods are either people look at it, or they just take the top K SNPs. They take the top K SNPs. They also can take the conditional method. We'll talk about that. And there's this other method. We'll kind of cover all those. So let's kind of go over how each of these methods work. So the top K SNPs, what it does is you just take the top K. So in this case, let's say K equals 5. You would take the 5 highest SNPs here, and you would say that I think my causal variant is in that set. That's what top K SNP works. Unfortunately, that doesn't work very well because in this case, these are the 2 causal variants. Okay, and again, we know this from simulation, and it's looking at the reason I'm showing this example is to show how clearly unintuitive it is because the interaction between multiple causal variants and correlation structure can produce really unexpected looking results. Okay, so the problem is that it might miss the actual SNP, and it kind of also ignores the correlation, right? If things are correlated or not correlated, that should give you some information. And also, finally, how do you pick K? These are the problems that have no real good solution. So the other popular method is called the conditional method, and this is actually widely used. It's an iterative approach. What you do is you select the top SNP, and then you recompute the statistics conditioning on that top SNP. So then in your second round, so you say this is one causal variant, then you pick this guy, and then you find the highest next one, okay? And you repeat that process. And then that set, you say, is your causal variance, okay? The problem with that is as follows, is that when you're making this choice at the beginning, you're basically saying that the peak SNP is the causal variant. And in this case, it's not, right? Because these two are the causal variants, these two circles. So that you're already missing that. And actually, how often does that happen? Like if here, these are two regions where we just randomly picked one causal variant, one variant to be causal, and we simulated the statistics, and we saw how often that variant is the causal variant. And here, what we see is, and then in both of these situations. So if there's not a lot of LD, and then we look to see where the causal variant is ranked. We just repeat this many times. So we see if there's not a lot of LD, the causal variant is ranked as the top SNP most of the time. But if there is a lot of LD, there's many times when the top SNP is not the causal SNP, just by chance. So there's a lot of, so here there's almost nothing, and here there's many, right? So if there's LD correlation, just sometimes the top SNP just is not the causal variant, just by chance. So the way we approach this problem is by basically giving up and acknowledging that it's impossible to find the causal variant because of this, right? But on the other hand, what we could do is if we try to do something that kind of loosens the problem a little bit, we could do something with some accuracy. So our idea behind our approach is that we come up, instead of trying to find the causal variant, we want to find a causal set. And the causal set is, we call it the row causal set. The causal set is a set of variants that contain all of the causal SNPs with at least probability row. So the idea behind this is like we're coming up with a confidence interval. Okay? Typically, we set rho to be 90, 95%. And we're gonna, and so we don't guarantee that all the SNPs in our set are the causal set, but we kind of we want to guarantee that the causal SNPs are in that set. Okay, so for example, a trivial solution is just pick all the SNPs in the genome as your causal set. Okay? If you do that with 100% probability, your causal variants will be in your set. Okay? Obviously, that's not a good idea because that gives you no information. So the idea is that you want to find the smallest number of SNPs that you're picking, smallest set that satisfies that property. So I'm going to show some examples of this before we get to how this works. So if I go back to that other example that I showed, the causal set that Caviar returns is these 7 SNPs. Okay, so it's these 5 guys and then these 2 over here, right? And it actually captures the causal variance, right? Because that's what these 2 guys are, right? Why does it pick these guys and not these guys here? It has to do with just the LD structure. So it knows what it's doing. And so it's saying that with 95% probability, it's going to be in these 7. So that's the idea. So how does Caviar work? So the idea is that, let's say we have a region with m SNPs. There's really 2 to the m possibilities of which SNPs are causal and which ones are not causal, right? So one possibility is nothing's causal, one possibility is everything's causal, and there's basically 2 to the m total possibilities. And we could think of this as a causal set is kind of like a binary string of zeros and ones. It's 0 if it's of size m. If it's 0 is causal and 0 is not causal and 1 is causal, right? And so for each causal status, so we think of there's 2 to the m possible causal statuses, and so for each causal status, we can compute the likelihood of the data that we observe. And then a causal set is going to be the subset where we think that the causal variants are in. And let's say that causal set contains k SNPs. there's really only going to be 2 to the k causal statuses that are consistent with that causal set. Why is that? Because if we're saying that the causal variants have to be in the set, there's only 2 to the k of them, right? So everything outside of the set has to be all zeros. So these are the two, kind of the causal set and the causal status, right? And for each of these status, we can compute the likelihood. And then here is a very kind of crude representation. But so given this region of M SNPs, given a causal set with k SNPs, kind of the posterior probability in quotes, right? That a set will contain the causal SNPs can be estimated with essentially this, which is the sum of the likelihoods of all the causal statuses consistent with the set, which is the 2 to the k terms, which are the possibilities that only the SNPs inside the causal set are the causal variants, divided by all of the possibilities, right? And if this is close to 1, that really says that the other possibilities don't have a lot of likelihood. And so we wanted this to be at least 95%. And then we need to find a set that satisfies this property. Okay, so there's a lot of details that are omitted, like priors and stuff like that, but this is the key idea. Okay, so how do we compute the likelihood? Basically, it's pretty straightforward. Statistics follow a normal distribution with mean and variance if we use z-scores. And if you pairs of statistics follow a multivariate normal distribution, and their correlation can be modeled based on the correlation of the actual SNPs that you can compute. And essentially, you could imagine that the results of the association study I'm kind of going fast here, will follow a multivariate normal distribution with a mean that we know, with a variance covariance that corresponds to the correlation matrix, and a mean that really depends on the causal statuses. Okay, so this I'm kind of going quickly here because we started a little bit late, but I'm teaching a class right now, Mondays and Wednesdays, 2 to 4. So if you're curious in this, like we're covering this, I think next week. So okay, so okay, I gotta skip ahead. So anyway, so if we look again the other methods, there's a top case SNPs. We talked about that conditional method, which is just this iteratively picking the top SNP. And then there's one method that I mentioned earlier I didn't discuss, but it's called one post, Mahler et al. And they basically they're very similar to caviar, except for the difference is they're only considering the possibility of one SNP being causal, and not more than one SNP being causal. And okay, so how do these methods perform? So here is simulated data. If this is the kind of the recall rate, how often does within our set we capture the causal variance? So if we have 1, 2, this is the same region was simulated with sometimes this is 1 causal variant, 2, and 3, and we can see that caviar pretty much has the recall rate, which is very close to the row, which is what we expect. And the conditional method really kind of breaks down pretty quickly with more than one variant. And the one posterior, when there's one causal variant, it performs exactly the same as caviar, which makes sense because it's the same method. But when there's more than one variant, obviously it breaks down because it doesn't take that into account. So if we look again, also this is like under 2 causal variants, 3 causal variants. Caviar obviously has a huge advantage over these methods. So this is the basic method, and so now let's talk about how we apply it in some interesting ways. So one way that we are applying it is to combine EQTL and GWAS data. So the idea is that most GWAS risk loci fall in non-coding regions of the genome. So the hypothesis is that there are variants that alter the phenotype through their effect on gene expression. That's the kind of general hypothesis of how non-coding variants affect traits. So if a G risk loci is causal, then it should also be causal it should also be causal for the gene expression of genes, at least in a specific tissue, right? This is actually the underlying hypothesis of why we do eQTL studies and collect them. And so co-localization is basically the method to try to detect the phenomenon. The same variant is causal in both GWAS and eQTL studies, right? Well, if we have a method to find if the causal variant, we can just apply this twice and see what we get. So here's actually some examples of a co-localization. So here's actually a situation where this is a GWAS and eQTL. What do we see here? We see a really strong peak. We see one SNP that's clearly the causal variant in both of these. And so we would say that variant clearly co-localizes. So the same variant that drives both the association signal here and here. While in this case, it's actually look, the peak variant might be the same, but there's a lot of other stuff going on, right? So it's not as clear. It might be, but also might be. So there we would say that there's less evidence for co-localization. Okay. So why is this important? Well, we don't actually know when we have a GWAS variant what gene is it affecting. And what we can do is if we have a variant, and we can look at all the genes around that variant, and we can look at them in all the tissues. And the idea is that if we see something that's really high, for example, it looks like this variant, this GWAS hit, affects gene 4 in blood, then that might give us some clue about the mechanism of the gene. So in general, one of the main interests in co-localization is because it can be used to understand mechanism. Okay, so again, how we do co-localization, it's very similar to caviar. I'm going to kind of skip ahead. There's kind of a couple existing methods that others do this, and they don't take into account one variant. We do, so we perform a little bit better than they do. But actually, I'm going to kind of skip ahead. This is the result of what we get. So we apply this to some diabetes data using the GTEx, and what we were surprised is that we find that the GWAS variants really classify into 3 categories. One is variants that co-localize. Well, that makes sense because that's what we're looking for. One that are ambiguous. Okay, so we don't have a lot of power. But then we also found a lot of variants where there's a peak in the GWAS and a peak in the eQTL, and they were different. And this was somewhat surprising. And so we actually have a paper on this now. But the key finding is that a lot of the eQTLs and the GWASs have very significant associations with the same gene, but they're different variants between the genes. And so I think this actually really kind of makes us realize that our underlying hypothesis that the expression of the variants will be affecting expression of the genes, and that leads to difference in disease, phenotype is something that it's clearly more complicated. Why that result actually got us looking into another question, which could be an explanation of why we see this, which is looking for allelic heterogeneity. And so what do I mean by allelic heterogeneity? It's when you have more than one variant that's causal in the gene. So here's an example. So if I gave you this plot and I ask you how many causal variants are affecting this phenotype, right? You would say 2, right? Because there's clearly 2 peaks and they're not correlated. But the shape and the significance levels can really, in this case, give you an idea, and the LD gives you an idea that there's more than one causal variance, right? So this is what we call, when we see this, we call this allelic heterogeneity. So we actually extended caviar to also compute allelic heterogeneity. The way that it works, we also have this causal set. I'll just actually basically say, so the way that we do this is that we look at all the causal statuses with k causal SNPs, with k ones in that thing. There's going to be m choose k terms, and then divided by all 2 to the m. And so each result of this for every k will tell us what's the posterior probability that we have exactly k variants that are causal in that locus. And so if we see if k greater than 1 is a high probability, then that's evidence that there's allelic heterogeneity. So this is a method that can tell us if there's allelic heterogeneity, even if we can't find what the actual causal SNPs are. And we apply this to simulated data, and we show that it works really, really well. And the interesting thing, so then we apply it to GTEx data. So here's actually an example of one locus where when we apply it, we see that with very high probability, there's only one causal variant here. That's one example. But here's another example where there's a lot of LD, there's a lot of significant variance, but our method tells us that there's probably at least 2 causal variants. We apply this widespread to the GTEx data. GTEx is a consortium that collected expression data in like 50 tissues, and we see that if you plot how many genes that they have with the allelic heterogeneity, of course, against the number of individuals in that data set, we see a kind of a very kind of a linear correlation, which really suggests that there's allelic heterogeneity almost everywhere in almost every locus. The more individuals we see, as we see more and more individuals, we're able to detect it. Similarly, we looked at 3 blood data sets, and we computed allelic heterogeneity, and we found that there's a huge amount of overlap at the loci that have allelic heterogeneity. So that this structure is even in different studies in different populations. And same with skin as well. And finally, we applied this to GWAS data, and we find that like out of the 108 loci that were recently reported for schizophrenia, 25 of them demonstrate allelic heterogeneity, and 13 out of the 37 loci at HTL have allelic heterogeneity. So we actually think that probably more than 25 and more than 13 of them, it's just a matter of like as the studies get bigger, we'll be able to detect more and more. Okay, so in conclusion, which variant, so we're interested in this fine mapping problem. That's the which variants are causal, and also which genes are causal, how many causal variants are in a locus, and if the same variant is causal in 2 studies, for example, GWAS and EQTL. And so this work was really driven by Farhad Hormoziari, who's now a postdoc in Harvard in Alquist Price's group, and most of these papers have come out. The last paper just got on allelic heterogeneity just got accepted to American Journal of Human Genetics. This is a joint work with Bogdan's lab, and Gleb was a key contributor in this fine mapping area. And if you want to follow us on Twitter, or blog, this is where you can find us. Okay, thank you.